Hi everyone, welcome to today's session. This is Srinivas Rao, Assistant Professor, Department of Microbiology, Dr. BRR Government College, Jadcherla. Today we'll talk about fermentation process. Fermentation is another anaerobic pathway for breakdown of glucose, one that's formed by types of organisms and the cells. As you know well that we have been discussing about sessions on cellular respiration. Cellular respiration is a process through which cell generates the energy and it also synthesizes new components which are required for other metabolic purposes. As a part of cellular respiration, we have been discussed about aerobic respiration process and anaerobic respiration process. Usually, the aerobic respiration process occurs in all aerobic organisms which breathe oxygen, and where in cellular system, oxygen will act as terminal electron acceptor. Wherein anaerobic respiratory organisms, they do not require oxygen as their terminal electron acceptor. In spite, they cannot live in presence of oxygen. We call them as anaerobes. This is a special case with especially prokaryotes. We cannot expect in higher organisms. The organisms which belongs to anaerobic respirators are prokaryotes and archaebacteria. And we have other respiratory organisms which carry out a part of non-oxygenic pathways, that is fermentation process. It is an exclusive mechanism we can observe, especially in bacteria and fungi, especially in yeast. Fermentation is an age-old process known to the people because the people are aware of their applicative products which they have been using in their daily life. For example, we are eating curd, we are making dosa and idli, we have been eating bread, we have been eating cheese, butter, ghee, and we have many dairy products, and we have been drinking wine and alcohol, and we have been doing many activities. All these are resulted of fermentation process. Unfortunately, we don't know the process behind it, but it's a fortunate that after the invention of the process by Louis Pasteur, we came to know that this is the process called as fermentation. The fermentation was first defined by Louis Pasteur. He described fermentation as life in absence of oxygen. It can also be found as those biological processes that occur in dark and that do not involve respiratory chain with oxygen or nitrate as electron acceptors. As I told you, we have two respirations, cellular respirations. We have aerobic respirations, where oxygen will act as a terminal electron acceptor, but where in anaerobic respiration, and nitrate will act. Nitrate will act as a cellular respirative molecules. But where in fermentation, it lacks. There is no electron transport acceptors. In physiological sense, fermentation is defined as a type of respiration wherein both electron donors and acceptors in organic compound. When you notice and observe the whole fermentation process, the process of electron generators and electron acceptors both are organic compounds. But however, for industrial mycologist and biotechnologist and microbiologist, they have a different meanings because it's an applicative. Sometimes we call it as a biological process that occur in absence of oxygen, or sometimes we call it as a food spoilage. Sometimes we call it as a production of alcoholic beverages. Sometimes we can call as a reduction and oxidation processes occurs because of organic compounds. And sometimes we call it as it's an alternative means of producing power disposal. Whatever may be the different meanings that the fermentation has, but the fermentation has merits. But the fermentation process is one of the important process very useful for the mankind. Which organic compounds will transform, and the same organic compounds will accept and donate the electrons. When you look up both the cellular respiration process, especially in aerobic and anaerobic respirations, you can observe the process of glycolysis and tricarboxylic acid cycle, and where the produced reduced components like NADH and NADH NADH and FADH2 both. FADH2. All these compounds, yes, FADH2. We have FADH2 and we have NADH2. These compounds which are produced during the process of glycolysis and tricarboxylic acid cycle accepted in the electron transport chain, as we have seen in aerobic and anaerobic respirations, especially oxidative and nitrate and sulfate respirations. But it's a special case with fermentative organisms. They don't have a system because they lack cytochrome complexes, they lack iron sulfur complexes. If you say another way, the bacteria or the yeast which have the fermentation process, they lack the electron transport chain machinery. When you observe the prokaryotic cell, the cell membrane consists of different compounds and complexes, protein complexes. They accept the electrons and they donate to the nitrates or oxygen. But wherein fermentative bacteria, you couldn't notice any kind of these membranes in the plasma membrane. Hence, they cannot carry out the process of electron transport chain. Then what happens? The so-called produced NADH2 and FADH2, again they should kick back again for the conversion of glucose into the pyruvate. So it is one of the beautiful process carried out by many microorganisms. And when you notice the physiological concept of fermentation, it's different. When you see now, this is the process of glycolysis where you can see what happens. You all know that the process of glycolysis, where does it occur and what occurs? You see the glucose. The glucose is converted to pyruvic acid. During this process, it produces the NADH2. NADH2. We all know that our cellular system has a feedback mechanism. We all know that our cellular system has a feedback mechanism. We have a feedback mechanism. What happens in during this feedback mechanism? Feedback mechanism, simple mechanism. For example, I'll give you a very simple example. If you are hungry, when you feel hungry, we'll keep plate full and we started eating. We eat and eat and eat and eat. At a particular point of time, after 5 minutes or 10 minutes, then what happens? Our belly says it's filled. Then what do? We stop eating. Or we slow down the eating. At a particular point of time, though our plate is half empty, then we stop eating because our belly says yes, it's filled. Then after 2 hours or 3 hours, again our belly digestion process carry out and belly will become empty, and then it says again I want food. What is this? When the belly is filled, it says it's filled. Then when it get emptied, we feel hungry, then we need to have it. The same process. When the process is taking place in a particular system, for example, this is the particular environment or system where the fermentation process is carrying out. When if you see here, when the fermentation process is carrying out in this particular environment, then what happens? The glucose must be converted to pyruvic acid. When the pyruvic acid is going to synthesize in the particular system, at a particular point of time, the pyruvic acid concentration increases in the cellular system. In this particular system, so we can notice the presence of pyruvates. You can see the pyruvate in this particular well. We can see the pyruvate in the particular well. So now it has a pyruvate in it. When the pyruvate is more, what is the fate of glucose? Glucose conversion must stop. Glucose conversion must stop here. From glucose to pyruvate, there must not be a conversion from glucose to the pyruvate. Why? This process we call as feedback mechanism. When this particular environment is completely filled with pyruvic acid, now the pyruvic acid must again it should convert it to some other product until unless the glucose cannot occur. This mechanism we call it as a feedback mechanism. So what need to do? There are some kind of organisms, they convert the pyruvate into ethyl alcohol, lactic acid, or butyl alcohol. Then it occurs a process. That is, here there is a oxidation. Oxidation takes place. Oxidation takes place. So when you notice the process of entire glycolysis process, in entire glycolysis process, we expect one thing, that is when glyceraldehyde 3-phosphate, when glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate, where on addition of inorganic phosphate, and this process of reduction takes place, and the reduction is taking place. The process of reduction is happening. So because of the reduction process, because of the, so now here a reduction is taking place here. Now this is the, here the reduction, NADH2 molecules are coming. When NADH2 molecules are coming, here is the NADH2 molecules. When NADH2 molecules are coming over here, then what happens? Here the NADH2 molecules. The NADH2 molecules concentration has increased again here. This, the concentration of NADH2 molecules and the concentration of pyruvic acid stop the conversion of glucose into the pyruvate. Then these molecules must be used up again. Again, there is a process. When a pyruvic acid, it depends upon the organism which is involved in the process. It completely varies. There are different microorganisms. Depends upon their enzymes which they have. Some microorganisms can convert it into ethyl alcohol. Some microorganisms can convert it into lactic acid. Some microorganisms can convert it into butyric acid. Multiple fermentative processes are there. Means the fermentation have a ecological significance. In aerobic respirations, carbohydrates completely oxidized and ATP and carbon dioxide and water will be released. But the fermentation products such as alcohol, organic acid, carbon dioxide, hydrogen are in that, they are being produced. It's a one of the important process occurs because it requires. So here, according to it, depends upon the end product produced during the process. We have different kind of fermentative processes. So there are many kind of fermentations. They named according to the end product which is formed. We can say it is alcoholic fermentation. We have a lactic acid fermentation. We have propionic acid fermentation. We have mixed acid fermentation. We have butanediol fermentation. We have butyric acid fermentation. Like this, there are various fermentation processes occurred because of the microorganisms involved here. And the crucial compound for conversion into different molecule is a pyruvic acid. Pyruvic acid, it's a key compound for all type of fermentations, because the only process is glucose must convert it to the pyruvate. From pyruvate, it will move on to the different paths, depends upon the organism. For today, according to your syllabus, what you have, we have two different type of fermentation processes. One is you have alcohol fermentation, another one you have a lactic acid fermentation. So I am going to brief you about ethyl alcohol and lactic acid fermentation in this presentation itself. After the formation of the pyruvic acid, exclusively there is a single step, there is a two-step process occurs in the conversion of the ethyl alcohol. One is, what happens is that this pyruvic acid is a 3-carbon molecule. It can undergo the reduction. When it undergo the reduction in alcoholic fermentation process, it can convert it into the acetaldehyde. When it converted to the acetaldehyde, again this acetaldehyde converted to ethyl alcohol. It's a one of the very simple process here. This pyruvic acid is there. The pyruvic acid formation occurs when the pyruvate is formed. Pyruvate where carbon dioxide is going to remove from the pyruvate and it converted into the acetaldehyde. Here there is a step here, and the pyruvic acid converted to, after release of the carbon dioxide, means in the one carbon is going to loses from the pyruvic acid and it converted into the acetaldehyde. Then acetaldehyde undergo the oxidation, where the so produced NADH2 molecules again they oxidizes and forms again NAD place, and they converted to the ethyl alcohol. We require an enzyme called as alcohol dehydrogenase. we require an enzyme called as alcohol dehydrogenase, and we get the ethyl alcohol. This is carried out by yeast like fungi, and we have a Saccharomyces cerevisiae, especially yeast, and when it comes to the bacteria, we have Gymomonas. Gymomonas are the bacteria which can carry out this process. And we need to learn about one of the important topic over here is a Pasteur effect. Pasteur effect is simple. Yeast, while growing on glucose anaerobically, obtain a yield of 2 ATP molecules for every 1 mole of glucose, which is much lesser than the ATP production under aerobic conditions. In order to cope up with this situation, large amount of glucose is broken down to obtain necessary energy. When the yeast are transferred from anaerobic to aerobic condition, the utilization of glucose is drastically reduced by the factor of 3 to 4 within concomitant stoppage of alcohol production. This phenomenon was first observed by Pasteur, and his honor it called as a Pasteur effect. Apparently, the Pasteur effect is a result of a difference in cell energy change under aerobic and anaerobic conditions. So basically, there is a Pasteur effect happens with irregularities in the energy conversion because of the shifting of aerobic to the anaerobic conditions. And alcohol fermentation is the domain of yeast. Some bacterial species also carry out the fermentation. The bacterial species opt for different pathways, and end products also vary both qualitatively and quantitatively. Especially Gymomonas mobilis, isolated from plaques, the fermenting juices of agave and metabolizes glucose via 2-keto 3-deoxy-6-phosphoglucoside, that is ED pathway, and splits pyruvate into pyruvate decarboxylase and to acetaldehyde into carbon dioxide, and acetaldehyde then produces the ethanol. And the Sarsinia vetriculae is another bacteria like this. And there are different kind of organisms which are present and which can carry out the process called as alcohol fermentation. It's a very simple process. The process is very simple. Formation of the pyruvic acid, and pyruvic acid directly converted to the ethyl alcohol, and there is oxidation process takes place with the involvement of the alcohol dehydrogenase. And we need to take care of the environment, and we need to take care about the environment ships, and we need to take care about the raw materials what we provide, and we need to take care about the switching of the process from aerobic to the anaerobic processes. It is one of the crucial process carried out by the most of the yeast and alcoholic microorganisms. At the same time, we have a lactic acid fermentation. When you notice lactic acid fermentation, especially in lactic acid fermentation, we have different types. We have homo-fermentative and heterofermentative, and we have bifidum pathways. So whatever it may be, the process it is carried out by lactic acid fermentative organisms. There is a peculiarity, lactic acid fermentation process. This is one of the process where it occurs in muscles. Especially it was observed in the muscles. When you do strenuous exercises, our muscles get strained and we feel pain. Then our biochemists notice that it is because of the formation of the lactic acid. When there is a continuous strain to the muscles, when you do rigorous exercises or running or jogging or any work, then there is a non-availability of the oxygen or oxygen consumed completely in the muscle region. There it leads to the short anaerobic conditions. There those the called pyruvate can be converted to the lactic acid. The presence of lactic acid in the muscles will give you a short-term pain that we feel it as a normally pain in the muscles because of the overstraining. And it was noticed later, it was carried into the blood and sent to the liver, and liver again it will convert it to the pyruvate back again. It's one of the physiological process usually occurs in the muscles of a higher organisms. First time the formation of lactic acid which is noticed later, the same process observed in the microorganisms which they are during the process of curdling, the milk turns to curd, and we have noticed the process of formation of the lactic acid. So lactic acid is very common end product in many bacterial fermentations. Some bacteria generally produces large quantities of lactate as an end product, which are often referred to as lactic acid bacteria and are collectively assigned to the family Lactobacteriaceae. And lactic acid bacteria are heterogeneous morphological and include rod, cocci, and various within these morphological types. The bacteria exhibit motile and mostly saccharolytic and excrete large quantities of lactic acid as a fermentation end product. And they have they do not contain hemines. However, hemines, because they cannot carry out the electron transport chain. Lactobacillus is one of the important bacteria according to the human health point of view. It present in the gut of the human beings. It exclusively present in the vaginal walls of the women, it present in the especially gut and vaginal walls, and protect the human beings from the most of the pathogenic and opportunistic pathogenic microorganisms. So that's why lactic acid is one of the probiotic bacteria which relies in our human body. And as I said, lactic acid fermentation is of various types. It's a one-step fermentation process from pyruvic acid. The pyruvic acid is converted to lactic acid simply by an enzyme called as lactate dehydrogenase. Then there is a single step where the reducer molecule NADHL kick back to NAD plus, and then it produces the lactic acid. Directly the pyruvate can be converted to the lactic acid, and this process we call it as a homofermentative process. Wherein simply heterofermentative process, we get ethyl alcohol and lactic acid, both the molecules. In heterofermentative process, the microorganisms undergo HMP shunt pathway, and where they produce almost all like HMP shunt pathway, they produce lactate and ethyl alcohol molecules. Both they can produce during homofermented pathway, and in bifidum pathway it can produce the molecules called as lactate, acetate, and it can produce ethyl alcohol also. So like this, different components are produced with the different fermentative processes. So fermentation, when you see this here, thymine pyrophosphate, notice here, and when you notice here, this is the difference between the lactic acid and alcohol fermentation, wherein it produces alcohol and releases carbon dioxide, but it produces lactic acid and does not release any carbon dioxide. There is no release of the carbon dioxide during the process of lactic acid. It is a two-step and a single-step process, as I said, and it requires magnesium ion and alcohol dehydrogenase, but it requires an enzyme called as lactate dehydrogenase with a Zn plus. It forms acetaldehyde is an intermediate component, but there is no intermediate component you can see in a lactic acid fermentation. It commonly occurs in yeast, but it occurs in bacteria, some fungi, and vertebrate muscles also, as I said. And when you notice here, according to energy kinetics, we have two different things. When you notice glycolysis and fermentation, glucose will be converted to pyruvate, and this is a pyruvic acid converted to either lactic acid or ethyl alcohol. It takes place in the absence of oxygen or presence of oxygen, but it exclusively takes place in the absence of oxygen fermentation. Here net yield of 2 ATPs, but there are no ATP molecules. Here reducer molecules are produced, but these are here reducer molecules are consumed over here. But when you notice entire production of energy, either in anaerobic respiration, especially in fermentation and glycolysis, you have a total 8 ATPs over here, but in fermentation you will get only 2 ATPs. The energy generation during the process of fermentation is very less, but the end products which are produced are very applicative and useful for the microorganisms, at the same time human beings. That's why fermentation is one of the exclusive process is associated with bacteria and some of the microorganisms. It has its greatest importance in producing. And when you see the industrial microbiology paved a new way. It's all because of the isolation of various fermentative microorganisms. When you look up, most of the life science industries are working based on the fermentation process. Today there are many products are being produced by the process of fermentation. We have production of cheese, production of wine, production of alcohol, production of industrial beverages, production of solvents, production of enzymes, production of vitamins, production of vaccines. So what not, we are producing many components which are required for daily usage of the human life and animal life on the surface, and we are producing many components all because of the fermentation process. Fermentation process is a user-friendly process taken up by the microorganisms, and it is one of the cheap process, but it gives a worthy elements and products which are very useful for the human beings. And fermentation process is one of the versatile process which is satisfying the hunger of the needy with a low cost. That's why fermentation process is one of the important process and one of the aero cellular respiration. But when we are going to see with respect to the cell point of view, cell gains very less amount of energy, but it gives very useful products which are useful for the other organisms. Certainly, these are very important components produced by the microorganisms. That's why fermentation process is one of the cellular process occurs in the absence of oxygen, and this is one of the important process uh, carried out by especially only microorganisms. Later sections will discuss about rest of the processes. Thank you.